In this video tutorial, I am going to discuss about digestion and absorption of carbohydrates. The major portion of our day-to-day food is the carbohydrate, and among the different carbohydrates that we consume, they are made up of starch, sucrose or lactose. So starch, sucrose and lactose. If someone is consuming non-vegetarian diet, then instead of starch, they are getting glycogen as a major source of carbohydrate. Now we need to understand that how these different carbohydrates are getting digested in our body. So the digestion of carbohydrate, it starts in the mouth. In the mouth, we have different salivary glands which secrete one very important enzyme, that is salivary alpha amylase. Salivary alpha amylase. This salivary alpha amylase, it is secreted by the salivary glands, but the major portion of the salivary alpha amylase, it comes from the parotid gland. Parotid gland. This parotid gland is one of the major salivary gland of the mouth, right? This salivary alpha amylase, it is activated by the chloride ion, activated by chloride ions. And if we talk about the action of the salivary alpha amylase, it breaks down alpha 1,4 glycosidic bond. It breaks alpha 1,4 glycosidic bonds. Now to better understand that how salivary alpha amylase acts, let's try to understand its action on the starch. So we know that starch is the polymer of glucose. So suppose this round represents glucose molecule and this horizontal line, it represents bond between glucose molecule. So this is the polymer of glucose, right? There are branches also. So we show 2 or 3 branches in the molecule of starch. So this is the molecule of starch, and similar bonding pattern is also seen in the glycogen, right? Now all this horizontal line, it represents alpha 1,4 glycosidic bond, except at the branching point, this bond. So at the branching point, they have alpha 1,6 glycosidic bond, and rest all bonds are the alpha 1,4 glycosidic bonds. So this salivary alpha amylase, it can break down all this alpha 1,4 glycosidic bond, but it cannot break down this alpha 1,6 glycosidic bond, right? The second limitation of the salivary alpha amylase is that it cannot break down this terminal glycosidic bond, right? Although it is alpha 1,4 glycosidic bond, but as it is a terminal alpha 1,4 glycosidic bond, salivary alpha amylase cannot break it down. So you can say that this bond can be broken down, this bond can be broken down, this bond can be broken down, but this particular alpha 1,4 glycosidic bond, it cannot be broken down by this salivary alpha amylase. So let's try to understand that once this starch is broken down by the salivary alpha amylase, what type of products we can get, right? So suppose this bond is broken down, then we get this 2 glucose molecule bound with the alpha 1,4 glycosidic bond, right? So let's write that first. So these 2 glucose molecules which are bonded with each other by alpha 1,4 glycosidic bond, we call it as a maltose. Then it is possible that this bond is broken down. So at that time we can get a 3 glucose molecule with 2 alpha 1,4 glycosidic bond, right? It is like this 3 glucose molecule with alpha 1,4 glycosidic bond, we call it as a maltotriose. Maltotriose. Then it is also possible that this bond can be broken down, this bond can be broken down, and this bond can be broken down. So we get 2 glucose molecules bound with alpha 1,6 glycosidic bond. So the third product is 2 glucose molecules which are bound with each other by alpha 1,6 glycosidic bond, and we call it as a isomaltose, right? Now apart from these 3 products, what could happen is that salivary alpha amylase, as it randomly breaks down this alpha 1,4 glycosidic bond. So let's say this much chain is removed, from here this much chain is removed, from here this much chain is removed. So what we ultimately get is the trimmed structure of the starch, right? So we can get such trimmed structure of the starch, and we call it as a oligosaccharide. Oligosaccharides. Now these oligosaccharides, they are generally 4 to 10 monosaccharide long. It contains 4 to 10 monosaccharide subunits. And second, that most of the bonds in this oligosaccharide, they are alpha 1,4 glycosidic bonds, right? And as we know that it can retain certain this side chain or certain branches. So some bonds are also alpha 1,6 glycosidic bond, right? So these are the products that we get by the action of salivary alpha amylase. But here there is one more caveat. What is that? See, this salivary alpha amylase, it is secreted in the mouth through the various salivary glands, right? And it has a limitation that it is optimally acting when the pH is equal to 7. But what happens? Within a very short time, food reaches to the stomach from the mouth, approximately 5 to 10 minutes is there, right? When food reaches to the stomach, at that time pH is less than 2. So at this much low pH, this salivary alpha amylase cannot act. So what will happen? Whatever action this salivary alpha amylase has to do, it has to be carried out in the 5 to 10 minutes, right? So because of this time limitation, not all the starch which is present in the food is able to get digested. It is estimated that only 20 to 40% of the starch which is present in the food can be digested by the starch. So remaining 40 to 60% of the starch, it remains undigested, right? So we can say that at the end of salivary alpha amylase activity, we get maltose, maltotriose, isomaltose, oligosaccharides having 4 to 10 monosaccharide residues, and remaining starch 40 to 60% undigested, right? So this is the action of salivary alpha amylase. Now the food from the stomach, it goes into the duodenum. In the duodenum, two important secretions from the pancreas, it mix up with the food, right? So what are these two important secretions? So the first one is the bicarbonate, and the second one is the pancreatic alpha amylase. Pancreatic alpha amylase. Now this bicarbonate, what this will do is that it neutralizes the acidic stomach content which is reached into the duodenum, right? So it brought about the neutralization. And second secretion, that is pancreatic alpha amylase, it is almost similar to the salivary alpha amylase, similar to salivary alpha amylase. That means it is activated by the chloride ion, it breaks only alpha 1,4 glycosidic bond, it cannot break down terminal alpha 1,4 glycosidic bond, it cannot break down alpha 1,6 glycosidic bond. So in each and every aspect, this pancreatic alpha amylase is similar to the salivary alpha amylase, except for the one thing, that is this pancreatic alpha amylase, they are more powerful. And now this pancreatic alpha amylase, they will be active. Why? Because the bicarbonate ion had neutralized the acidic pH which was brought about by the stomach acid, right? So now what this pancreatic alpha amylase will do, that whatever undigested starch is there in the food, that will be get digested, and everything which is related with the starch will be converted to maltose, maltotriose, isomaltose and oligosaccharide, and no or very little undigested starch will be present. So if I want to write down the summary of the product that we get by the action of pancreatic alpha amylase, it is maltose, then maltotriose, isomaltose and oligosaccharide. Oligosaccharides of glucose homopolymer. Now from the duodenum, food reaches into the proximal part of jejunum. The proximal part of jejunum, suppose this is the jejunum and this is the lumen. The proximal part of the jejunum, it is lined by the enterocyte having a brush apical border. So, suppose this is the cell lining the jejunum. Its cell membrane which is facing towards the lumen, we call it as a apical membrane, and the cell membrane which is not facing towards the lumen, that means this membrane, this part and this part, we call it as a basolateral cell membrane. Okay. So, this apical portion which is of the wavy line, that is brush border, this brush border, it is bound with certain disaccharides. So, these are the different disaccharides which are bound with this brush border of the proximal jejunum cells, right. So, what are these enzymes? So, there are numerous enzymes which are present. The names are lactase, maltase, sucrase, isomaltase and oligosaccharides. So, these are the different from that earlier salivary and pancreatic alpha amylase. See, salivary and pancreatic alpha amylase, they are secreted inside the lumen. They are not bound with anything, okay. Whereas these enzymes in the proximal jejunum, they are bound with the cell membrane, okay. Here one more thing, that this sucrase and isomaltase, these are of course two different enzyme activity, but if you see the structure of this enzyme, these are not two different enzymes, but they are part of single protein. Suppose this is the single protein. The one part of this protein, it has a sucrase activity, and another part of this enzyme has a isomaltase activity. So, basically sucrase and isomaltase, these are two different activity, but they are part of single protein. So, we can call these as a dual role protein, right. Now, what are the actions of these different enzymes? So, see earlier from the digestion of starch, we got maltose, isomaltose, maltotriose, oligosaccharides. These are the products that we derived from the digestion of starch, but we can also consume directly maltose, right. We can also consume directly lactose also. We can also consume sucrose also. So, whether it can be derived from the previous larger carbohydrate, or we might have consumed directly these disaccharides, then they will be digested by this different enzyme in the proximal jejunum. So, the maltose is broken down by the enzyme maltase and it gives rise to 2-glucose. Isomaltose, it can be broken down by isomaltase enzyme and it can again gives rise to 2-glucose molecule. Maltotriose, here this one is tricky because maltotriose, it can be broken down by the same enzyme, that is maltase. So, maltase can also break down maltotriose and can give rise to 3-glucose molecule. Oligosaccharides, they are broken down by oligosaccharidase enzyme and it can give rise to multiple glucose molecule. Lactose is broken down by the enzyme lactase and it can give rise to glucose plus galactose. And sucrose is broken down by the enzyme sucrase and gives rise to glucose and fructose. So, here in the proximal part of the jejunum, all the, whether it can be polysaccharide, it can be disaccharide, all of them are digested completely and ultimately they are converted into these monosaccharides, right. So, we can say that digestion is complete over here, and at the end of digestion, and if we consider the general food that we take routinely in our diet, it is estimated that more than 80% of end digestion product of carbohydrate is the glucose, right. And about fructose and galactose, These are the approximately less than 10% each. So, generally these are the monosaccharides that we most commonly get as a end product of digestion. Now, once digestion is complete, these simple monosaccharides, they need to get absorbed through the intestine into our blood, right? So, there are, for different monosaccharides, there is a different rate of absorption. It is generally estimated that rate of absorption, it is higher for the hexoses than the pentoses. And if we consider these different hexoses, the highest rate of absorption is for the galactose, then comes glucose, and then comes fructose, right? And as I had already told, that hexose is higher rate of absorption than the pentoses. So, at the last it comes pentoses. So, this is the comparative of different rate of absorption for the different monosaccharides. Now, we need to understand that how these different monosaccharides are getting absorbed. What is the mechanism of absorption? So, galactose and glucose, they share a common absorption mechanism, that is sodium dependent secondary active transport. Sodium dependent secondary active transport. This fructose is getting absorbed by the facilitated diffusion. And pentoses are absorbed by the simple diffusion. Let's understand all these three mechanisms one by one. So, first I will start with the sodium dependent secondary active transport mechanism, and by this mechanism glucose and galactose are absorbed, that is sodium dependent secondary active transport. Now, this absorption, it is also taking part in the jejunum and some little portion in the ileum. So, suppose this is the enterocyte. On this side there is a lumen. This is the cell which is made up of which makes the absorptive surface of the jejunum and ileum, right? And on this side we have interstitium. Now, as I told you earlier, that this portion of the cell membrane which faces to the lumen, we call it as a apical cell membrane, whereas this remaining cell membrane, it is called as a basolateral cell membrane. So, on this enterocyte, on this basolateral side, there is sodium potassium pump. This sodium potassium pump is the active pump. That means it requires ATP. This pump, it will send 3 sodium out of the cell and 2 potassium inside the cell. So, as this pump is continuously working, what happens is that as the sodiums are moving out of the cell, there is very low sodium concentration, right? Now, on this apical cell membrane, there is one transporter is present, which we call it as a sodium glucose transporter 1, that is also its short form is SGLT1. This SGLT stands for sodium glucose transporter. What this transporter does is that it transports sodium and glucose from the lumen to inside the cell, right? From the lumen to inside the cell. Now, this transporter is not directly utilizing energy. See, why? Because this sodium potassium pump, it is already bringing down sodium concentration very low inside the cell. And so, as sodium concentration is very low, there is a concentration gradient for the sodium from outside to inside the cell, right? So, along with this concentration gradient of sodium which is brought about by the sodium potassium pump, glucose also moves. So, we can say that this SGLT pump is not directly utilizing energy or ATP, but it is indirectly utilizing ATP, right? So, if suppose this pump is inhibited, then this pump cannot be working, right? So because of this, we call this transport mechanism as a secondary active transport mechanism. Now let's try to understand that what will happen if glucose is entering inside the cell. So after some time, glucose concentration will start increasing, right? Why? Because the glucose is continuously being transported from the lumen to inside the cell. So now there is a concentration gradient from inside the cell to the outside of the cell. And here very strategically located transporter is there. We call it as a GLUT2. GLUT stands for glucose transporter. This is passive diffusion, right? So this GLUT2, it transports glucose from inside the cell to the interstitium side of the cell as per the concentration gradient of glucose. And this is how glucose is getting absorbed through the intestine. The same mechanism also works for the galactose, right? Now there are certain important inhibitors of these various transporters are there. There is one important inhibitor, that is fluorescein. This fluorescein is found in the unripe fruit and bark of the apple tree. It inhibits binding of the sodium to this SGLT1 transporter. The second inhibitor is vabain. This vabain is the toxin. It was traditionally used as a arrow poison. It inhibits this sodium potassium ATPase pump, right? And the third toxin is the cytochalacin B. Cytochalacin B. This toxin is found in different fungus and it inhibits this GLUT2 transport mechanism, right? So this is about the how glucose and galactose are getting absorbed in the intestine. The second mechanism that we have to talk about is the absorption of fructose, and it occurs by this facilitated diffusion. So let's try to understand that how exactly the fructose is getting absorbed. So fructose is getting absorbed by the facilitated diffusion. Again, let's say this is the enterocyte. This is the apical cell membrane and this portion is the basolateral cell membrane. That means lumen is on this side, this is the cell and this is the interstitium, right? Now fructose, it is either absorbed by facilitated diffusion. So here it is facilitated by GLUT5 transport, GLUT5 transporter. This transporter, its name is GLUT5, and here again GLUT stands for glucose transporter. So it transports fructose from the lumen to this cell. Okay. Now from the cell to this interstitium, different books says different mechanism. Many standard textbooks says that glucose is transported from inside the cell to the interstitium by the same transporter, that is GLUT5. Whereas some book says that it is transported through this basolateral membrane from the cell to the interstitium by the transporter which is utilized in the glucose transporter, that is GLUT2. GLUT2. If you ask me, I will be agreeing more with this transporter mechanism. Okay. So, but both transporter are suggested by different standard textbooks. So, you can remember any or both of these transporter mechanism. The last transporter mechanism or last thing that we need to discuss is the pentoses. That how pentoses are getting absorbed. So pentoses, they are getting absorbed by the simple diffusion. Simple diffusion. So simple diffusion is very simple to understand. Again, this is the enterocyte, this is the apical cell membrane, and this remaining is the basolateral cell membrane. On this side we have a lumen. So pentoses, without utilizing any transporter, they are directly absorbed inside the cell, and from inside to this interstitium side they are directly absorbed, and there is no any ATP utilized. So we call it as a simple diffusion. So this completes digestion and absorption of carbohydrate. In the next video, I will discuss about the disorders of digestion and absorption of carbohydrate. If you have any query or confusion, please write it down in the comment section below. Thank you.